Hi everyone, my name is Amy Lee and I'm a rising junior majoring in cell biology and neuroscience with a double minor in history and public health. This summer, I had the incredible opportunity to conduct research within the Department of Neuroscience and Cell Biology at Robert Wood Johnson Medical School. Under the direction of my PI, Dr. Michael Matise, and my lab mentor, Lita Wong, I was able to complete the project titled Tracing Lineages of Sonic Hedgehog Responsive Cells in the Developing Central Nervous System, as summarized in this poster. Sonic hedgehog is a ligand morphogen that plays a crucial role in cell fate determination in developing embryos. In the canonical pathway, sonic hedgehog binds and inhibits the patch receptor on the cell surface, which then allows a protein called smoothin to become disinhibited and activate downstream effectors, including Gli1. During early neurogenesis in mice, from about E5.5 to E8.5, where E0 is the time of conception, sonic hedgehog is produced ventrally in both the nodal cord and the floor plate, and it diffuses to generate a concentration gradient along the dorsoventral axis of the neural tube. Differential exposure to sonic hedgehog causes cells at varying positions along this dorsoventral axis to adopt different lineages. 5 progenitor domains, P0, P1, P2, PMN, and P3, are established along the dorsoventral axis by combinatorial expression of paired box homeodomain proteins, which is illustrated under the introduction section. The project I worked on this summer sought to trace these lineages of sonic hedgehog responsive cells in the spinal cord of embryonic mice from E8.5 to E10.5. We used mice with the genotype Gli1 Cre-ERT2 heterozygous and Rosa26 flux TD tomato. This means that all cells that transcribe Gli1, which again is a downstream effector of sonic hedgehog signaling, will also express Cre-ERT2 recombinase. This Cre recombinase acts to excise the stop codon located after the Rosa26 gene when tamoxifen is administered. This ultimately allows for transcription of fluorescent marker TD tomato, which can thus be used as a marker for Gli1 expression and sonic hedgehog responsivity. Tamoxifen was administered via oral gavage to the pregnant mother at E8.5, E9.5, and E10.5 in order to observe the distribution of cells responding to sonic hedgehog at those times. E0 was estimated to be the time in which a vaginal plug was found after mating. Embryos were then harvested at E12.5. Tissues were fixed with 4% paraformaldehyde solution, washed with DPBS, mounted in OCT, and frozen on dry ice. Tissues were sectioned with a cryostat to a thickness of 14 microns, then stained with DAPI and NuN. DAPI stains cell nuclei, while NuN stains neurons. Our results are shown in these images. As shown, TD tomato expression at E8.5, which is shown in red, is widespread all along the dorsal ventral axis, but its concentration decreases towards dorsal areas. By E10.5, TD tomato is sparsely expressed in ventral regions, while virtually disappearing from dorsal regions. Overlap between TD tomato and NuN expression also decreases from E8.5 to E10.5, but notably remains in area P3. These observations were made while taking into account that the effects of tamoxifen may last up to 3 days, and that TD tomato is strongly expressed even in cells that transiently express Gli1. We can thus conclude that sonic hedgehog responsivity in the spinal cord generally decreases from E8.5 to E10.5, and it also disappears entirely from dorsal regions, most likely due to lesser concentrations of sonic hedgehog. The decrease in overlap between NuN and TD tomato expression further indicates that sonic hedgehog-linked differentiation of cells into neurons primarily occurs before E9.5. Of course, this project will continue to expand in the future. Next steps include quantifying changes in TD tomato expression, quantifying changes in overlap between TD tomato and NuN expression, staining tissues with other markers to identify lineages of unidentified sonic hedgehog responsive cells, and analyzing tissues at later gestational dates. To finish off, I would like to take a moment to thank everyone that supported me throughout this experience. Thank you to all the members of the Matise Lab for your guidance and patience throughout this summer. Thank you to Project Super and Nicole and Kayla, of course, for this amazing opportunity and for always supporting me in my journey as a student in STEM. Thank you everyone for listening.